If they change. My guess is that permanent instances where you get a fixed IP are probably really expensive at AWS.
I originally bought a server, got a server for AWS, like at the beginning of this year, and then I only uploaded once, just one web page, 15 KB, and then the next month they sent me the bill. It's 1400.
Oh my gosh.
For the usage of it. It's like a SFTP server, so I guess that's
Oh.
so I panicked online because it's I will never end up using it.
That's why I was talking to John Ray about this. You can do anything on AWS, but they have such a complex tiered
Oh, of
charter
the
model
payment?
for data uploads, data movement on different server
Yeah.
clusters, a billion different options for what CPUs you can have, what they have available. You know, I've been doing AWS for 10 years, and I still can't start a project or any clue.
I search online, the best advice I
And
got.
they, you'll find these tables that are like, oh, it's like 0.02 cents per minute. Well, and you look at those and you're like, yeah, but that doesn't jive with my $685 bill I got last week.
Wow.
And then you're like, oh, but you were on tier 2, so you're looking at the wrong one, you're gonna jump down here, and it's like, and how did I know that? Ryan Lair tells this story about when he was a postdoc, they were working on this large shared data resource that was like a bunch of human data, so it had to be HIPAA compliant and everything.
It's 1:09, just to let you know, in case, because I know you have your phone call. Okay, okay. I didn't know if you had to call.
This AWS thing, and what he ended up doing is he logged into a different node and pulled 6,300.
Oh my gosh.
Because
For just pooling?
into that node, he would move the data from tier 2 to tier 4. They were able to call and basically go,
Oh, that's good, but
but they basically said when they call, we will not take, we will, your account's been marked, we will not forgive increment charges. He goes, I have no way to know.
yeah.
Oh, right, so that,
Wow.
well, one command,
I'm just ignoring that message of the bill due, and then there's no suspect bank account. Just get email address.
How do you turn this mic?
the mic is controlled here, it is on. You can turn the volume up, it also turns the gain up
Perfect.
slightly.
Okay. I'm like, my voice doesn't always project.
This thing should work, but we haven't got it to work for this, and I don't know what the problem is.
Okay. Yeah, I'll give it a few minutes. Oh, yeah. I'm like, I have my charger, but if you have yours here, I'll just make sure I don't accidentally take it, because,
It's a pro, right?
yeah. Yeah, it is a pro. But I was just like, I can see myself just packing and taking it. All right, I don't. I'm like, I have the opposite problem, Sam. I'm so short, so I'm like, I don't even have to bend. Do we have about 38-ish? 1, 2, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 17, 18, 19, 20, 21, 22, 23, 24. Okay. We have about, 25 people. So after 5 more people come in, then I'll start, I think. Just because I'm like, we have a lot. And last year, I remember we didn't finish. No, last year, I think we had to do the IGV tomorrow. So, you're teaching it tomorrow, so I guess it's not bad. Like, we were able to do that in the assessment with no problem, but yeah. So I think we don't finish today, we can take it over tomorrow. But we'll see if we can actually finish it today. No, this is just what Mary put in the capstone, so. I feel like I'm not the right one to talk about. This is like Rob to talk about it and explain.
Yeah, she has such a
Yeah, I would just be like, her husband drew this. For the longest time, I didn't read this when I first joined the lab, so I thought they just took it from Game of Thrones. Like I didn't know, understood why they took this map from Game of Thrones and stuck it onto our website until I actually read it one day, and I'm like, oh. So- okay. All right. I think I'm gonna go ahead and start. Can you guys in the back hear me? I have, I think I can hear echo, so I can hear myself projecting. I'm gonna go ahead and start because we have actually quite a bit to try to do today. So what we're gonna talk about today is we're gonna actually work with actual data today. So we have some data files on the server that you guys are actually gonna pull down, and we're actually going to go through fast QC. So we're gonna look at the QC, do trimming, actually map it, and then try to visualize it today. So there's quite a bit, and we're actually going to, for homework, you guys will be taking these files and taking them all the way through to BAM because we'll be using them next week when we talk about like RNA-seq and other processes. So I'm going to give you guys kind of an overview of what we're doing today. This just kind of gives you an overview of what, in general, you tend to do with this data. So you're going to get from the sequencer these FASTQ files. And one of the first things you typically want to do, if you recall, like when Lynn was talking, is you want to do some QC to make sure that these files are worth actually moving forth with. So we'll kind of do a fast QC. There's a lot more programs that we'll be introducing to you guys next week that allows us to do some further QC as you run other programs. But the first one we're going to go through today is fast QC, and that's a nice way to see whether or not the quality of the fast Qs, whether or not they're good quality, as well as after we do some trimming to try to remove the adapter, whether or not actually remove the adapter. So we're going to go from the fast QC, and we'll be doing some trimming, and then we're actually going to map it to whatever the reference genome that you're using in your experiment. In our case, the files that you look at today will be human. So we'll be mapping it to reference human genome. And then once we go from there, once you have these map files, that's where you can actually start moving forth with a lot of other softwares and programs that you can use to kind of look at differential expression, to kind of visualize it, to do different modeling. For today purposes, we will take you from the FASTQ down to the actual mapping. We'll talk more about these files later on, but you'll produce these binary files that are called BAM, and then from there, we're gonna take it into the X2Go and the IGV. So we kind of introduced you guys to that the first day, so we're gonna actually visualize them on the IGV. So that's kind of like our plan for today. So I think Mary included this in the slide, so she wanted to remind you guys that typically most tasks will take about one node, and the best way for you to decide whether or not you need more than that is typically to actually look at the manual, and in the manual see whether or not it talks about parallel processing. So like Trimmatic that we're using today can actually be parallel process, so you can request more than a node, but the keywords that you're typically looking for to kind of decide is in the manual itself whether or not it has these parallel processing, the number of CPUs that it recommends, NTAS or processor. And if you are requesting a certain number of nodes within your SBAsh, based on the program you're using, so right here, for example, I'm using this generic program and it's actually giving a flag for how many CPU, just make sure that they match. So here, if in the program with itself, you're requesting a certain amount, just make sure that in the SBAsh, when you define it, that you also are requesting them same task. That way, when Slurm is actually reading through your SBAsh header, is actually reserving the right number of CPU for you. So today, because we're going to be doing something that's a lot more, it's going to require a lot more tasks in memory. This will be the first time that you guys will see traffic jam, and by that, that means that some of you guys, if people are being greedy and requesting too much, some of your programs won't run and you'll start getting kickoff. So I guess the point of the slide is to get ready for some chaos. If it does happen, just put your red flag on. We'll come over and we'll try to figure out what's going on. And just a reminder, the sticky notes, does anybody need the sticky notes today? I'll put one here to pass on this side. This way, so yeah, as we're going through this, if you have any questions, just put like the pink/red sticky notes on. And if everything looks good as we go through and you finish certain tasks, because we'll be doing 4 different tasks today, then just put a green sticky note so that we know that once I see the green sticky note, then I can move to like the next step that we're doing. All right, so, oh, I didn't introduce myself, by the way. I'm Jessica, sorry. I forgot to mention my name. I just was like, we gotta start. Okay, so I am gonna go over some, let me just, all right. So the first part of today is just doing the QC and the trimming. So again, so you guys were already introduced to this yesterday when you wrote your first, second, third, whatever S batch. So today I'm gonna just walk you guys through that again, but this time give you a little bit more guidance as far as how I typically write my script and the reason why. So, you know, everybody has like, you'll find like what's comfortable for you as far as how you want to organize, but I'm going to walk you through how I typically do it. And the main, I think the one thing that I saw yesterday was that, I think Mary mentioned this too, is that you guys just had your files like all over the place. Like some people had them in their home directory, some people had them in specific directory. So one of the main things as you start processing your data is trying to figure out a way to track all of that. So that's kind of like what Mary hinted on that she wanted to cover in the beginning of today. And you want to be able to keep track of that. Otherwise, like as you generate more and more data, it gets really easy to start getting lost and figure out where your files are. So having like a system in place, having your scripts documented well is a nice, so that you can trace back, is a nice way to go about that. So I have a worksheet. So if you go to the website, in addition to my slide, you go down, I think it's under worksheet. There should be one that says FastQC and trimming there. So when we get to that, you can kind of follow along. Okay, so what we're going to do is we're going to look at our FastQ files, and we're going to try to determine the quality. We're going to look at the raw file first, and then kind of see whether or not we can understand what is telling us with regards to quality. You'll see that it's a little difficult to read, so we're going to actually use that the software that you used yesterday, the FastQC, to actually generate something that's a little bit easier for us to interpret. And then from there, we'll do some trimming. So so if you are looking at the PowerPoint, just notice that I crossed out the blue because I wrote this all before we uploaded everything to the AWS. So the path is just a little bit different. So I'm just putting that red. But basically, what you're going to do is you're going to actually look at the FastQ file right now. So just, I'm going to just emphasize this. These are the raw data, your FastQ. So we do not want to edit these FastQ, especially because the FastQ right now is on the server for everyone to view. So if one of you guys vim it, then it edits it for everyone. And these are raw files, so we never want to edit it. So you're going to use more or less head, but don't use actual VI or vim on it. So typically what you want to do is when you do get your raw file from the sequencer, put it into a folder, change the mod so that you can't actually edit it, and then you can copy it down into wherever it is that you're processing your files to actually go through the process. But just make sure that you don't edit your raw files, because I don't know. If you do that, then I don't know what to tell you. Okay. All right. So you can go ahead and log into the server right now. If you go to this, right here, this path, you'll see that there are 2 FASTQ files there. We can just open them up with either more or less, or head or cat, or one of those, so you should view it. But this is what you will actually see, is that you'll see these lines of code. If you look at them, you can see that there's about 4, the pattern is about 4 lines for each one. And each one of these lines, what they basically stand for, your FASTQ, is the first line is just a descriptor. This is gonna tell you information such as the sequencing information. So like what lane it's in, what sequence in. I think it gives you information about the adapter. So that's that first line, it's just a descriptor. The second line, I mean, that should be pretty recognizable to you guys, that looks like is probably your sequence. And then the third line is just this optional descriptor. Typically, most of the FASTQ file I see, it has a plus sign in it. And then the last line is this quality score. This is going to be alphanumeric score. So basically, it's going to rate the quality on, it depends on your sequencer. Typically, it's FRET33, so it's like a log 10 base. So because it's FRET33, that means like 1 in every 1,000, there's a chance. Like it's telling you the quality of like what the chances of error. So if you look at this, this offhand, I would say, looks like a relatively good quality. The reason why I said that is because it's mainly alphabet. The more symbols you start seeing, where it starts looking like cursing symbols, the more likely that's a bad sequence. So it's basically yelling at you, telling you, don't use me, because it's cursing at you. So if you see that on line 4, that's already like a flag that maybe you shouldn't move forth with this. Yeah, so I just answered my own question. I should have asked you guys that. But yeah, so this is how you can tell the quality. But this right here, can you imagine if you look at the thing, if you do a WC, HL, and see how many lines there are, you'll see that you have quite a bit of lines. And for you to actually go through this and try to determine the quality of every read, so each one of these is like a read, that would take you forever. It would be hard to do it, which is the reason why we're going to actually use the FastQC. So just a reminder that if you have poor quality to begin with, yeah, your data, you're going to have poor quality coming out. So it's going to, so we want to make sure that we actually work with good starting product.
And if you ever need the red scores for any reason, you can just look up tables online.
Oh, yeah.
So 0 to 40, 40 is good, 0 is bad, and you can just look up the red table.
Yeah. I think it's like 33, and the other one's like 66. It's like the devil? No. But okay, so this kind of, this will make more sense once we actually see the FastQC. This is in the slides, so you can kind of go back and look at it, but basically, this is what the FastQC report is going to give you information on. So it's going to give you information on like everything from the GC to the adapter to any type of duplication you might have in your reads and sequence quality. So let's go ahead and actually start doing the FastQC so that we have those files to look at. So I'm going to exit out this real quick and just, so I have a worksheet. I'm going to go through the worksheet. Yeah, let's go through the worksheet. I'm trying to see, and then I'll start making notes about stuff like module load and stuff as we go through it. Can you guys read that from the back? Okay, so the worksheet, I'm giving you guys the 2 links to the actual manual in the very front. And so as we're moving through this, the paths might have changed as we go through it. This was, when I tested everything here, it was on Fiji. So for example, the compute is going to be a little different, like right, we use like the partition compute versus like short and long. And then obviously the user is going to be different. So just keep that in mind as you're looking through this worksheet. So one of the first things that we want to do is typically we want to make some working directories. before we start our FSKC. So we know where the data path is. These are the files, these are the folders that I typically make. So I typically make one that's going to be my input. Like if I'm going to copy any data over, I have like an input folder, I have an output folder, I typically have a script folder, and then so yesterday, I think like a lot of people just had the error and output move straight into their like home directory. For me, I typically have an error and output folder that's within, what I call, every single time I run like a set of code, I call it an experiment. So I typically have it specific to my experiment. The reason why I say that is that depending on the bioinformatics software you're using, a lot of times it will place information that is important for the scripts that you're running into either the output and error, and they're not always intuitive as far as where they place it. But if you imagine if you had that in your home directory, it's gonna fill up really fast, and it's gonna be really hard if you actually need to go back and get information. Like I know some of the software puts information about how it trims into, I think it's like the standard out. So if you actually need to pull that information out, if you have it in a kind of like a big folder, it's just gonna be hard to go through. So I typically create it for myself in my experiment folder. So why don't we go ahead and kind of start off with that and just do like a day 4 folder and make the directory. So you can call it bin or script, however it is that makes the most sense to you for how you want to organize. So again, for me, it's typically an error output folder, an input, an output, and a script folder. That's how I typically organize it. I'll have you guys do that, bro. Oops, this is, yeah. I was like, why does it keep saying traumatic? Sorry, thank you. That's my fault. I wasn't looking at my thing and I just saw traumatic and I thought maybe it was an error on my part. Okay. And another thing I think everybody's kind of used to doing this is that I use PWD and LS a lot because my memory, I feel like sometimes like a goldfish, so I can never remember where the hell I am. So I'm always like reorienting myself by putting PWD and LS. And another reason why I use PWD a lot is that I saw a lot of you guys typing things out earlier in the week and not always using tab. I think the most common mistake is typos. So I'm always PWD and just copy and paste that into my script, whatever directory that I need or want to call on, because like it's not worth spending an hour just to realize that, oh, you mistyped your name. And that's the reason why your scripts didn't work and didn't put it into the right folder. So I use PWD a lot, not only to figure out where I am, but also just to copy and paste it. So does everybody kind of have, like, put, I guess, the green flag on if everybody has their directory and I can kind of move forward?
Is everyone on a cluster?
All right. Okay, so that's, yay, so we finished that. So if you want to kind of make sure that your folders are actually where they're at, then LS, like that's usually what after I create all the directories, I always LS, make sure, and PWD, make sure they're actually in the folder directory I want, and I actually have the folders that I wanted. So the next thing we're gonna do is we're actually gonna locate our FASTQ files. So because so you can CD into it, I typically don't CD into it because I just want to see if they're there. So I believe that I'm already on cluster. Let me see, yeah. So I usually just, right now I'm just going to see whether or not I'm actually on or what files are there. Okay. So I see here that we have 2 FASTQ files. So now what I typically do, like you won't need to do this till you actually start writing your script. But this is like, for example, this is when I would just PWD, I didn't CD into that, sorry. That's probably why I told you guys to CD. So I usually will just do that and I will copy this whole thing because this is long. And if I type that out, I can pretty much give you money that I will mistype something. All right, so we made the directories. Oh, here we go. Sorry, wrong one again. I'm just going to close this one. All right, so now that we have it, let's start writing our SBash. So I have an example one that's in the script folder that I put together of how I typically write it. I guess you guys can start with that template. It's under our directory under scripts, and it's titled day4_fastqc. So you can start with that, or if you already have your template from yesterday, since keep in mind, like the template that I'm using here again was like it was all test in Fiji, so there might be some things that are a little bit different. If you already have a template that works, by all means, just go ahead and use that. But I'm going to kind of show you some of the things that Mary was talking about as far as how you want to write. So again, here we have our job name. Put something descriptive in it. This is mainly so that as you're tracking things, especially if you have a lot of software that you're running and you want to be able to track like what has to do with FastQC, what has to do with bed tools, what has to do with trim. If you have a job name there, it'll just be easier for you to keep track of. You want to, so I don't know if anybody mentioned this. Yeah.
So remember, do not edit that SBash script where it is. Copy that back
Oh, yeah.
into your local directory.
Thank you. Yes, yes. Don't edit it there, because then everyone's unless you want a bunch of email from everyone pulling down your email that you put in here. But yes, thank you. One of the other mail types that I'm gonna just bring up, because I don't think specifically was brought up, is that if you can either specify that you get that email. I'm sure that unless you guys are the type of people that didn't get any errors yesterday when you wrote your SBash, you probably got like some emails that you had designated as your email address. So you can put none, beginning, end, fail, or all. I think in the edit, in the SBash, by default, we put all. But I typically, for me, I put fail at this point, just because a lot of these scripts, I know what I'm expecting. So Julie, I want to know if I failed because I did some kind of error, like typo. Okay, and then I'll put an error right here. I want you to just specify this one. So this is the standard out, and this is your standard error files that is going to happen. This is not the same as the output file that you create in your actual script. So like, for example, yesterday we did the FastQC. That's the output file that's specific to FastQC. The output here that you're specifying your SBash is your standard out and standard error. So that's usually something that I want in my error and output folder. So I think there was a little confusion there, so I just wanted to distinguish that those are two different output. And this this percent sign, X and J, this actually coordinates with the job name. I think on the AWS we can't do that. I think, that's my memory from last year. So I think if you do a %X, it will actually type %X. But the %J will actually give you the job ID. So every one of your error and out, if you look in your EO folders today, your error and output folder, you'll notice that there's usually, if you have the %J, there's usually like a number. That's the same as when you said SBash in your script. It designated a number to you. That's your job ID. So you'll see it there. So that's another way for you to go back and look at your error and output folder. Yeah.
What's the difference between my .out file and the output of my analysis?
Wait, hold on. Your .out file and the output. So this is your standard. You mean like what is the difference in the standard output versus?
The actual output of like a pipeline working.
Yeah. So like, for example, FastQC, what was your output file there that you guys were aiming for when you ran that? Yeah, that HTML zip file, so the actual FastQC. So that's designated by the software that you're using. Your standard out is more of just a log that you're going to get of information. So a lot of times that'll either be by whatever bioinformatics software that you have. Like sometimes they'll have a specification that they'll push to your output or your error file, but it'll also be whatever you want to type into your output. So I'm going to show you just real quick right here. So for example, I have this print job that I usually have here, and mainly I want this in my output just so I can keep track of like where was my in directory, where were my out directory. So this is all going to be pushed into my standard output, like what I have here, and it's mainly because I want to be able to keep track. I don't always do it very well, but I'm attempting to be organized here by having this here. And it'll tell me like where was, like what cluster was run on and so on. So that's kind of what's in your output. And then depending on the software, like for FastQC, we see that in our standard error file, it will actually keep track of like FastQC, but that's not true for every software. That's specific. So it's pretty specific to whatever software it is. And it usually will put in their manual what type of output file and what they put in it. Yeah.
What I would say is that I saw a lot of people yesterday have an error folder and
Oh,
an
yeah.
output folder, and they stuck both their .out files and their FastQC output in the out folder. That's probably not best practice.
Yeah.
Normally, you're gonna wanna name whatever your output is for whatever analysis you're doing. So usually we have like a one directory that's like an E&O directory, whatever you wanna name that. And then a separate output directory for the actual program.
Yeah.
And that way you know if you're actually looking for the stuff that you care about, if it worked. If it didn't work, then the out and error files are for troubleshooting. If it works, then you want to know where that's going, and you go see in your directories, you see FastQC output, oh, that's where I'm going.
Yeah. Does that make sense to everyone? The time that I go into my E&O is to make sure things are running properly. If otherwise, I'm going to go straight to my actual output directory that I specify as output within my experiment. Okay, so I think this was introduced in the, day one, this idea of doing variables. I'm going to reintroduce it here because it starts making a lot more sense why we were teaching you guys these different path variables. So today, for example, we're going to run two different programs within one script. And so I'll be referring to my input directory and my output directory a lot to simplify and the code so it's easier to read, as well as to make sure that I don't have typo. I'm going to go ahead and specify whatever my path is in my right here in my in directory. So usually I have a section that says set required variables, and I'll have like an in directory, output directory. If I'm going to use the same file name over and over again, I'll put like a file name. So this is the variable name, this is the actual path. As when you type this out, just to save yourself some grief, I usually will then ls whatever this path is, again, just to make sure it actually exists, because I feel like that, like from just typo, that's a really good chance that you'll have that. So you'll want to, this is, again, a preference, but it's really, I would recommend it because it's just going to make your code a lot simpler. All right, so this is pretty much the same thing that you guys wrote yesterday, but I'm going to explain it a little bit more. I'm hoping everyone can see that. Let me zoom in a little bit more. So this is the last part which is actually running the job. So I think yesterday, if you don't specify, for example, an output directory, it will just put it wherever. It'll put it pretty much wherever you're running your script. So we'll put it into the script folder. So one of the first thing I do typically is I do a make directory -p flag here. I don't think we specifically cover that. The reason why you have that is that if you don't have the directory, then it pretty much just, I don't know, it sends it into like space or something. Like you won't be able to easily find your files. If you have the directory and it already exists and you are telling to make that directory, it will balk at you and tell you that directory already exists. So you have this flag so that if the directory doesn't exist yet, it will make that directory. If it already exists, it will just ignore and won't remake. You won't have two directories with the same name. So I would put that there because a lot of times like you're thinking like, where do I want these files? And then just in case you didn't do that in the beginning, when you were making a directory, this just makes it so there's somewhere for your files to go. FastQ, so here I'm calling, I also module load up here. I forgot to put down. Module load, so we told you what to module load yesterday. If you don't know, like for example, I know I want to do FastQC. I don't know what version we have. So you can, I usually do module spider FastQC. So once I type that in, module spider is basically looking to see if it can find that software and what version it has. So here it tells me that we have 2 different versions. And then if I want to know how, like if you don't know exactly how you want to module load that, like I'm going to load this version, then I will, if you do the same one, the same command, it will then tell you this is exactly what you need to put in to be loaded directory. So then I usually just copy and paste that into my script. So I already loaded the module up here, and I think this is the first time that you guys might have seen these backslash and stuff. I think we mentioned it, but I'm gonna mention it again. So here, so let me just walk you through what's happening. So I'm loading this module, I'm telling it, okay, print all this information about what I'm gonna do, make this directory, and then run this actual FastQC. If you don't have the module loaded, which you guys all did this yesterday, so this should be familiar, when it reaches this line, it's not gonna know what it is that you want it to do. So it's just gonna error right there. I have, for readability, this is a really small script, so that it's really easy to read. But for readability, I usually do is this backslash or forward slash? Backslash, thank you. The backslash, and then you notice that there's a color change here of yellow. If you see that it's still green, you probably have a space in the back, and then it will error on you. So just make sure you see that shift in color that tells you that it's actually registering as a backslash. And then I will put in my in directory. So this is, I'm telling you, this is my input file that I wanted to run. This star, again, is a catch-all, so it's just gonna go through every file in this in directory that has this ending and look for it and run the program through those. You can specify one specific file as well, yeah.
So just one point about backslashes, there's kind of 2 reasons. I know that a lot of you are kind of getting frustrated about like long lines in Vim, the fact that you have to go back and forth, and you can't just use your cursor. So backslashes are one way to split up your lines so that you can actually go to specific arguments in your command without having to go through, 80 characters of text first. So that's one reason to use backslashes. The other is to split up the arguments in your command so that if FastQC has, the command is FastQC, then it has parameters, then it has, you need -o output directory, and then you need your file. You can split those all up into different lines so that it's just a little bit better organized in terms of readability.
Yeah, I would agree with that.
Those are 2 reasons to use backslashes.
Yeah, so you can see this is a lot easier to read than if I had like the whole, and then this is also, because I use these variables, also easier to read because I'm not having this long list of path, which can get by doing this, you're just simplifying it so that if it does error, you can go back real quick and fix whatever error is. And then -O is for output. So here I'm gonna give it the actual out directory that I want it in. So again, this is all kinds of stuff that we didn't really cover yesterday, and we just kind of let you guys add it. If you run it today, it should make it so that you can actually see that you can actually start getting how you want to go about getting yourself organized so that you can, run all these scripts. So let's, you have a question?
If you're doing, like, specifying the output here, do you need to do it in the S-batch, like, at the top?
No, the S-bash, remember, is just to talk to the node. So that S-bash header is more to tell it, like, what resource do you need, and the reason why you have your standard out and error is because that's part of just collecting, like, that's part of, like, the whole process of running the program and figuring, like, if it does have an error, like, where it's going to put your error in. But Everything under.
Standard output directory?
It's like when we're writing all those S-dash lines at the beginning of every script and we're having an error in an output.
Right, so this is a different output. So this is kind of what I was talking about.
Yeah.
The .out file is different from whatever program you're running. So think of .out files as .out files, not output. They're not the output from the program. They're just .out files. They're for troubleshooting. So,
Yeah.
from the major D. Right.
So like the dot out is going to be whatever the computer prints out on the screen while it's running the program. So if it's saying program start, program finish, that'll go into your dot out. But the actual output of the program, you know, the file that it's working on, the directory that you specify in the command itself.
So anything that's in the command that's an output has nothing to do with the .out file. I know it's a little confusing, so,
Yeah.
but output means two different things.
Yeah, one is more of a log of like, yeah, the computer talking to you and telling you what it's doing and what the errors that is like seeing as it's running the program, and the other one is your actual software that you're running itself.
So basically, actually, you can think of it as it's the out in the header, and then anything that's an output in your actual script is totally different.
Yeah. All right.
That it's spatially separated as well.
Yeah, it would be in the actual out, the E and O out, that's what I call. So yeah, so it would be that, because that's what it would normally print, just to let me know what's happening. So I'm just having it printed into my out file. All right, so let's go ahead and just run a FastQC on those files. Like, so just pick either one that's in that data directory, and we're just gonna run FastQC again, and then look at the actual FastQC report. So we'll stop there and then we'll move on to the next one once everybody has it. So, yeah, so let's run that SBash, and then if you hit any error, go ahead and put the red flag on. We'll, as helper, we'll walk around.
We're here to support you guys.
Is that clear, like what we're doing? Is there any question, or? I hope I'm being clear. Yeah, it's data file, they warn you should choose FastQ file. Yeah, it should be one, right?
You're doing great. She's doing it. I am one push. I am back.
Oh, yeah. Yeah.
really one of the making us push. Anyways, now the question, what were you saying? Or you can just, yeah, copy it first. No, yeah. You copy it. Right. With the. Mine starts with whole.
So, we'll see.
Yeah, the whole thing is just file off your file. And always into the same folder. The other thing you can do is mark outside. So you can select through all these. There.
That's the problem.
Alright, you found it. Okay, then you can, yeah, you can, I would scroll up. Yeah, just add the name.
Yeah.
And then go back to details.
Those are just.
Yeah.
So, I'm supposed to let me just go.
Yeah, now you just gotta contact me. If we're real good, we might need it.
So.
Yeah.
So I guess they just. So you're gonna have to change that.
Yeah. Do whatever. Yeah.
It's the right data, it's traumatic.
Oh, no, actually, no, I think. Right, the same name. Yeah, I mean, I don't think you should name it. Naming is removed. Naming is
Maybe some of the flags are funny.
same name. And we need to. Alright,
So make sure that that's just.
yeah.
So the Kappa prep kit that I got from. You can do.
Yeah.
I didn't know. So I did well.
Me on road.
It's probably somewhere.
minutes later, job later, and the whole time I just feel more ecstatic. But you know that hey, every time, what is this? So it doesn't matter if it's 10 minutes or 15 minutes. That's pretty straightforward. Yeah, why is it? Sorry, I just switched over, yeah.
My best friend, my mother.
Aggressive now, okay. What's the gift you have? Oh, yeah. Yeah, it sounds difficult, I'm sorry. What's that? I didn't read off the directory.
Yes, and this person is my
I heard they have used last names for duplication.
Oh, yeah, it's very right. You've been called it. Around the past few years. You just made it very. To make it very. Oh, okay.
So today, it's just radar. It looks like you're just making that scratch.
but every time I try to run
Yeah, what is your arrow?
because I can't my error output and then it said
So today, all the way to like 2:30.
this is where there's something
When our stations don't finish.
okay, so I'm taking it out of this source to run this
I didn't know about that. Yeah, because I have it is extremely the lights my license.
So it looks like you have to make it. Yeah, your script's never gonna start. Yeah, burn down your house. It's like, just move into the woods. Bury yourself. Okay, if you're making it, you got this. Just make sure
Yes, it's either on the dark side. You should check out your house in your area. Here, yeah, so they would be in this spot. I don't know, it would be where you specify it should be here. You got where you said your out files. Can you copy that and then I think I can figure out from that everything like this. Yeah, I also can't read it, but just copy that and put that in your see what else. Yes, you're going to make that critical. Yeah, because we have to do that. Like our is you need to find two directories as well. So you don't have ABC instead of yeah. There you go. And it works all right. It's high. I got a guy. Yeah. Like, I saw it. Especially because, most of the time, earlier when you got is RSD and Jennings is telling you
if you are able to get to the point where you have like a file, do we need to like go over how to rsync it back to your home directory and everything, so that you guys can view it? Because I'm gonna kind of just go over. Sorry, I just realized I have this on. I'm gonna go over kind of like the fast QC report, but I want to see where everybody is. So are you guys at the point where you're ready to like download the actual HTML file? If so, would you please read you guys can see whether that is enough to read? Yeah, if you have the HTML file, you're ready to rsync it.
If you have the HTML output, put your green sticky up, please. Thank you.
I think it's like half the room. I'm just trying. Thank you. Yes. I think so. Okay, you guys, I'm gonna go real quick on how to just rsync. You guys have been like copying on the server from one directory to the other. If you want to copy it on, because your output is gonna be HTML file, so in order to view the HTML file, we have to actually open it on whatever browser that you want, but we want to take that down to your local directory, so basically your local computer. So if you are on the actual compute right now cluster, you're not gonna be able to point it to your home directory. So you have to either open up another terminal window or log out, and then you're gonna use that rsync command. The dash R is just a flag to restrict it, but that's, but basically you wanna put whatever host, the whatever file you wanna copy, and then the next line will be wherever you want to copy it to. So here, you're gonna have your actual user at our directory right here, colon, and then the path to your whatever file you want. So this is when I would just pwd to get the actual direct path and file. And then the next part will be on your computer. So you're gonna have to figure out what path it is and what folder you want to put it onto your computer so you can actually view it. So just keep that in mind that as you're pulling it down, you can't be on the cluster if you're gonna pull it down to your home directory. You need to be on, you need to log out and then be on your actual local directory.
We'll just make sure
lies in the actual state of the states and tends to for a long time, I was just like, I felt like I was just doing something wrong. So I just it's just like when we first started doing it's like I just wanted to finding the right amount. Exactly.
The adapters being removed too much.
Okay.
And so I'm trying to see, so let me just show you what we
Okay.
decided to do. As far, sorry, it's right there. So this is our actual, just what we had. We're doing pair end, because,
Leading, trailing. Okay.
yeah, so that's when we're choosing to run. And
So these are pretty long reads.
yeah, they're pretty long reads. So this is before, they're 100. After we did the trimming,
What's the...
huh?
Okay, hang on just
Okay.
a sec.
So after
So
we did,
290. So it did trim like a few thousand.
yeah,
4,000
them are
reads.
few of
Or
them.
it dropped 4,000 reads, which means that it's trimming something enough to drop those reads.
Yeah, I'm just wondering if we had to be more strict with our trimming, because it seems like we still weren't getting rid of the adapters that we expected to.
Well, go down to the bottom.
Yeah, I think we're gonna move forward. A few people
Yeah.
are stuck, but I think I see enough green flag
Very bottom. Very
to the
bottom.
Kama.
Adapter sequences. Oops. Yeah, so there's no adapters there.
Yeah.
It could be that your library didn't have a lot of adapter contamination. Like, there's nothing there.
Yeah,
So
I
that's
don't-
why it didn't trim much.
Okay,
In order to get, so
I'm wondering.
this stuff is just like do a leading edge of like 10 bases.
Yeah,
And
I
these
think,
are
I
long
think
enough
that's
that you
just
can do
the
that.
sequence read. Yeah, it's going on the sequencer, right? That's just.
Yeah,
I
and
think
sometimes I do
errors.
that if the reads are long enough. These are long enough reads that I
Yeah.
would totally trim those off. But that's just the
Like the head
the
there
left.
and
Yeah.
stuff. Yeah. So we could go. I feel like this is probably is adapted, so we probably could head it.
Yeah.
I think here. I mean, I won't on this one, but yeah, I think we did leading to
Leading,
3,
yeah.
so maybe we can just increase that.
Yeah.
Okay.
Yeah.
Yeah.
That way they would at least see something visibly different between the two.
And then this
Maybe.
one here, I think I talked to Mary a little bit, but I wasn't worried about this duplication level
Oh.
because it's.
No, there's.
I feel
It's
like
kind of odd. There's a lot that are like a few sequences, which is not something I see often. Usually, I see a lot that are unique.
Yeah, I
and
usually see
then-
more like
and then it like
a much more steep one.
like
and
I
I'm not
don't worry
seeing
about that bump and the fact that there's nothing greater.
Are you
isn't
worried
a
about, like,
problem.
this gradual?
I'm not sure I would worry about it. It's just kind of odd that there's a lot that have 5 copies. You know. But,
Yeah. I'm used to seeing a lot
This
more
is-
up here. This is the.
Yeah, so it'll probably be all right. It is one chromosome too, so
That's
the
true.
smallest chromosome, so you might see more duplication.
Oh yeah, I have to
in
keep that in mind.
general.
I forgot that this is one chromosome. So yeah, that's just my. Because we add more chromosome and stuff like that, we might see it.
Yeah, you're gonna see a little bit better complexity.
Yeah, I kind of forget that. Okay.
Okay, yeah. Do you want me to talk about the report in general? Or
Yeah, actually, yeah, you could talk about the report in general.
Okay.
I kind of was like trying to figure out, but I was like, after we trim it, I was like, I feel like I can cut more off, but I wanted to. I'm like, this is RNA-seq data, which I haven't worked with. I wasn't sure if this is like typical or not.
it doesn't matter that much.
Okay.
Usually the mapping percentages don't change a lot.
Okay.
But if you have 100 pieces, then it's like, why not?
Okay.
You might as well.
Yeah, because you still have enough data there. Yeah, I noticed they were also
If you have
really long.
37 bases,
Oh,
then you don't want to trim off 10.
Like all the stuff I've been working with are like usually 30 to 50 bases. I'm like, why is it still so long?
Yeah.
I'm like, but I'm not familiar with this type of data, so I wasn't sure. Okay, yeah.
Yeah.
So I'll have to talk about a report because I think you've looked at it a lot more than I have. Like, I've been
I've
looking
looked
at.
at tons.
I know. I've been looking at just my Pacific data, so that'd be great. Okay. All right, you guys. I'm gonna start moving forth because we still want to do a few more things today, so can I have you guys kind of focus back here? And we're going to be doing Fast QC again, so it's not like you guys aren't going to have practice running the Fast QC. Let's see. All right, so I'm gonna actually have Lynn, because she's been looking at a lot of these reports, but if you recall, like I told you guys to go ahead and put it onto your local server and open up to your browser. So I ran it, uh-oh. Do I have to turn it back on? I don't know what happened. Okay, there it goes. Okay, so this is the report that you should have seen. This is on the, I think the N1. These are parent reads, and we'll talk a little bit more about that when we actually go into Traumatic. But this is the report that, that you should be looking at on your browser. So I'm just gonna have her kind of go over the different section of it, and then I'll have you guys start running trimming to actually remove adapters.
Okay, so there's a lot of information in these reports. Some of the graphs aren't as useful as other graphs. There's kind of a few that I go through in a first pass, and then there's more that can kind of show more problems with your library. One thing that I always look at is this number right here, total sequences. That is the number of sequences that you got off the sequencer for that sample, so that is your read depth. If this is the raw data, it is your raw read depth. And so this is a subset of a larger experiment that is just chromosome 21. So the fact that there's only 300,000 reads here is not concerning. If there are 300,000 reads here and you are doing something in a mammalian and it's a full sample, then that is incredibly concerning. But it's fine for the smallest chromosome. All right, and then, so this has general statistics. This is the name of your FASTQ file. This is, the encoding is, if you Google this, then it will tell you that it's FRED33 encoding, and you can look at the table to see what the quality scores correspond to. Jessica went over that earlier. Total sequences, see if there's any that FastQC flags. FastQC actually usually doesn't flag a lot as poor quality unless they're really bad. It kind of depends on how your settings are. You might need to actually specify quality within, something within the FastQC command in order to get it to flag sequences as poor quality. These are 100 base reads, and then it tells you what your GC content is in general. So this is a quality. So many of these graphs are position and read. And again, I showed you a couple a few days ago that are also based on the position and read. And this is your quality score, general average quality score based on your position in the read. So this is all of your reads at the first position. This is your distribution of quality scores at that position. Yeah. A quality score is the machine's confidence that that base is correct, based on how it can discern clusters, based on what the intensity of the fluorescent signal that it sees. It's just its confidence that it's correct.
And it's worth noting that there is a standard for how to calibrate quality scores, but that sequencing machines and the companies that build them have a vested interest in that number being high. So they don't. The reality is they tend to be a little optimistic.
So in general,
So when it's bad, it's really bad. Okay.
So in general, as I talked about earlier, you're going to have base errors and you're going to have phasing errors. And in general, you're always going to see your quality decrease over the course of your read. And that's because of the error rate and of phasing, where it can't tell if more than one nucleotide is being incorporated. There's just more problems with the blocking, unblocking reaction. And so, and it's discerning of whether there might be polyseg.
As the sequence gets longer, there's more DNA hanging off, and so the distance away from the original glass slide is being longer. And so there are like physics constraints that limit how good it can get the longer the reasoning gets.
Yeah. So you're always going to see some kind of drop-off. Usually, this is a pretty decent drop-off. So yeah, these last couple bases are going down into low quality in some portion of the library, but that's all right. It'll be okay. So this is generally a pretty good quality, as you can see, based on them being in the green. This is an actual picture of the quality on the flow cell. So this can show you whether there's massive, just technical problems that have,
Scratch on the flow.
yeah, like a scratch on the flow cell, you know,
I've actually seen a fingerprint on one of these before.
a channel being partially blocked or something, you know, anything that happened on the actual flow cell that is obvious here, then that's something that you can go back to your sequencing facility and say, what happened here?
Many times this will just be a big blue field,
Yeah,
meaning everything was great.
yeah. Not especially, no. You'll see like large stretches. If it's concerning, you'll see large stretches of like green or red in the middle here. Normally, we don't worry so much about things, especially for reads that are 100 bases. We don't worry so much about things at the beginning or the end, because as you're going to see next, you can trim off the beginning and the end and still have plenty of read in the middle that will map to your genome. All right, so this is another quality graph that just shows how many, it's a histogram of how many reads have what qualities, what average qualities. Most of these reads.
Now taking every position in the read, averaging it for that read. The average quality score for each read, plotting that as a histogram.
So almost all of these reads are in the 30s. That's good. As I said, the scale goes from 0 to 40. If most of your reads are in the 30s, then your library is probably pretty good from the quality standpoint. So you saw this in your homework. I'm not going to go over this in detail, but you can see irregularities in base composition. You're almost always going to see something at the beginning or end based on, you know, whether you have some inserts that are too short and you're reading into adapters, or you're usually going to see that. You can also, especially if the reads are long enough, you can trim those off. Most of them will still map all right if it's only a few bases at the beginning or end.
Stay right there for just a second.
Yep.
You will notice at the top where it says per base sequence content, it's got a big red X. So for every one of these tests, FastQC tries to evaluate whether they're good or not. So you get green, orange, or red. But it is assuming you're doing DNA sequencing for SNP detection. So when you're doing other types of libraries, they'll also often fail these tests, even though there's nothing wrong with them.
Yeah.
So keep that in mind.
So for the most part,
Scenario, that 5' end bias probably comes from ligation, because ligation is not really actually random.
in terms of ligating the adapters onto the inserts.
And it is very typical in an RNA-seq type assay, so this one looks pretty good.
Yeah.
But he still failed this test, because that's not what you see if you were doing random DNA sequencing.
I never, ever look at the colors of these circles, because that's not what actually tells me the information about my library.
I agree with that.
All right, then you got a GC content. This is, the best thing that this tells you is if you have major contamination of some kind. Duplication, you can see a little bit further, but if you have like organismal contamination, if you have some kind of bacteria that is highly prevalent and is going to have a different GC content than the actual organism that you're sequencing, then you might see a big hump here if it's like a higher GC organism, and that's something for concern. If you have major duplication problems or enrichment problems where you're seeing the same sequences all over the place, this is also going to reflect that, but it's not going to tell you the base cause of that. Per base N content, N is what the sequencer calls when it literally has no idea what base to put in. When the quality of whatever it's reading is low enough that it doesn't have any confidence in any of the bases. So if you have lots of N's in your library, then that's a problem. But it's also going to show up in the quality metrics on at the top of this report.
It's also worth noting that modern sequencers, they actually filter the reads you get back, so the ones with really lots of ends, they just don't give you.
Yeah.
So this per base N content has gotten better with each generation of sequencing machine. It's not that those don't exist, they just figured out that they didn't give them to you, they look better.
Sequence length distribution on raw data on Illumina machines, you're always going to get one length because that's the length of read that you are getting off. That's what you paid for. For trimmed, so you'll see, should I show them maybe? So you'll do trimming in a few minutes. But you will see that this is not one number. There are some reads that have been trimmed somewhat so that there's some that are 74 bases now instead of 100. So that trimmed off, the trimming program that you'll run, trimmed off 25 bases of your read, and then you can use that 75 base read and then map. So it doesn't really matter for raw data, but it gives you a sense of what your trimming has done after you run trimming. Sequence duplication, again, I went over this a few days ago. Adapter content, I also went over. There's not really adapter content in here. This library actually probably would be okay without trimming. You're going to run trimming on it anyway. But it looks pretty good. It would map pretty well. There's another set here that this library doesn't have a problem here, but there's an overrepresented sequences where it basically goes along with the sequence duplication level. If there is any specific sequence, if there's 10,000 instances of one specific read, it will tell you what that read is so that you can figure out, is it a poly A stretch? Is it, and it also has its best guess of what it is. So if it recognizes an Illumina primer or adapter, it will tell you that that is what that sequence is. There's a lot of times it says no hit, in which case it could still be a primer or adapter. It could be something from a different organism. It could be a repetitive region in your genome that has just been, it could be ribosomal that has just been amplified a lot, but it will tell you, if you have a bunch up here where you have a lot of the same read, it will tell you what those reads are. So then you can troubleshoot if you need to go back and do something else with your library, whether you need to enrich or for your next library prep, what you need to worry about. Any questions on the FAST? Yeah. No, for some applications you definitely can do that. Yeah. It just kind of depends on what you're doing, and you need to think about your statistics as you do that. Anything else on FastQC? Cool.
So other than FastQC is not the only quality software, it's just one that's really easy for you guys to get running. There are lots of other QC options with these bad readings. There's a program called QC that runs lots of stuff. In the minimum, if you've not looked at a FastQC report, you shouldn't do anything else with the data. Full stop. I've had people come into my office, spend 2 weeks working on RNA-seq data and can't understand the results they get. You pull up FastQC, and the experiment doesn't work. Like, when you have to tell them that, there's tears.
And I put a link in the worksheet that actually links you to, okay, I don't know where it is, but it actually, my computer is not registering me. Sorry. I don't think so. It doesn't want to escape. Okay, well, until I can get it to escape, I'll just tell you. Like it's either in the worksheet or in the slide. I think it's in the slides. There's an actual link to the bioinformatics that just shows good fast QC reports and bad fast QC reports, just so you guys can have an idea. Like it took me a while, and it still does. That's why I didn't come up to like take a look at these if I'm not always sure. So one of the things that I also noticed, like, for some reason I might have to restart my computer. I don't know why it won't let me exit. Oh, wait, it's just let me do something here. Okay. I'm like, okay, well, one of the things that you'll notice after you do your trimming, so what we're gonna do next is actually trim our adapters off. And so you'll notice that it'll tell you like this number of sequences that have changed, because if any of the sequences are like too short, for example, it's going to drop those because you can't really map that. So that's one of the parameters that you can put in your trimmatic, is saying if it's below a certain number of length to go ahead and just remove those. And then you'll also see, this is actually a trim one. You'll actually see that the sequence length here is different compared to what I started with. So like right off the sequencer, everything was like 100 base pair, but then once I do the trimming, then you're gonna have variation. So I'm gonna go over, so I think this is the link that's in the slide right now. So if you wanna just take more of a look at what these FastQC files look like and what really bad data looks like and what good data looks like, then you can go and use that as a resource. So now this is what we're gonna do, is we're gonna trim. And let me just go back to, so basically we trim because we want to get rid of, poly A tails, any adapters. If they're too short, they won't map. If they're low quality, that's the reason why we trim. There's other things that sometimes you want to look with regards to the library, whether or not you actually have to flip the reads. So if it's supposed to be like on the understrand, and you might have to remove poly A. This is the library today. I forgot to mention in the beginning that what you're looking at is the reason why you have Lynn mentioned it, but the reason why you have so few is we only mapped it to one chromosome. So your data, when you look at your real data with the full chromosome, it just might give you a different distribution than what we're seeing in our FastQC today. But again, you saw this in the video, but this is just to remind you, these are the adapters that's going to be placed onto your sequence, just so that you can actually have the sequencer attaches to the flow cell and actually be able to read through. So that's what we're trying to remove, is these adapters, because they're not going to map to your reference genome. So that's why we want to remove them. So there's a lot of trimming tools that are out there. So the video that you guys watched was on Trimmatic. We're going to do Trimmatic right now because we're going to work with parent data, and there's many other trimming programs available, and each one has just like their different caveats. So you're welcome to explore and just figure out if there's one that works better for your particular library. But just to let you guys know, we're specifically looking at Trimmatic today. So I put in a few other ones that you can look up and kind of see if it makes more sense for your particular library. So we're going to module load Trimmatic into our S batch, and this is the code that we're actually going to look at. You have to actually point it. It's a Java software, and you actually have to point it and give it the path. And so so I would recommend using the S bash script that I started. You'll have to, as you guys noted before, that what I wrote on there was meant for Fiji. So you're going to have to make some changes in the header to make sure that it actually works on the cluster that we're working on, as well as just some of that other variable and pointing. Somebody pointed out that there's some confusion with regards to /user versus /scratch. I like to think of these as like completely different computers, basically. So we're doing everything on Scratch because it pretty much, at least on Fiji, is because it runs faster. I'm not too sure with the AWS, if that's the same deal, but you have your user directory, and then you have your Scratch directory. So everything that we prepare for you guys is all in the Scratch directory. That's how we're able to share it with you guys, because we obviously don't have permission to your own directory. So basically, I think the thing to keep in mind is just make sure that you pay attention to like what your path is, because I think people are pointing it to both Scratch and user, and you can technically work on them in a dual way, but you just have to keep that in mind, because there seems to be some confusion there. But, so in the Scratch under script, there should be a day, and day 4, there should be a day 4 traumatic, and I would just suggest copying that to wherever you're gonna work on your script, just because if you misspell anything here and after the Java, it's gonna have a hard time pointing it. So I just put it there, but right here, and as you can see with the Lumina, the Lumina clip, there's a specific path, and it's in the script that will point to the actual sequences that we're gonna actually trim. And then you can set a bunch of other parameters for what you want for your trimming. Again, this is gonna be specific to your library and what you want to have here. So here, I'm just giving you an example. If you go onto the Trim-O-Matic website, they will give you more information on what you can remove. So this is just an example of, there's more than these parameters here. But for example, you can do a sliding window. So basically, it just kind of goes through in chunks and kind of see whether or not it hits a certain quality. And if it's below a certain quality, then it's basically going to mark that and trim it. You can decide that you want to cut off the head or the tail. So for example, in the when we looked at FastQC, we saw that there was a lot in the head that maybe we want to trim. So, and I think in the program I defaulted to 3, but maybe you want to play around with that and see if you can remove a little bit more. If it comes, I would do that if it turns out you have an issue with mapping, then maybe you want to, maybe it's having a hard time mapping because you have more sequencing at the beginning. So these were really long reads. Depending on the type of library you prepare, your read length can vary. These ones were 100 base pairs. As you can see, even after we did the trimming, a lot of it was in over 99. So if you cut off a little bit more, you still have enough that it should be able to map. But these are just some of the examples. Min length is another one that I typically use, because if your reads are really short, there's just going to be a lot of error when it comes to trying to map, because it can map in multiple places. So usually I set a minimum length as well. But there's a link in the slide that you can go to for the manual that will just give you more specification. And these are going to vary from trimming program to trimming program. So just make sure that if you choose to do like Trim Galore, for example, that you look up their specification. Okay, so we haven't done the trimming yet, but I'm just going to show you, because I did the trimming a little bit earlier, just because everyone's working at different paces. I'm going to go ahead and show you a little bit more of why we run the FastQC again afterward. Because, let's see. I don't know where I am. So I told you I'm always like PDing, so where did I put it? Hold on, I don't even know. I can't remember where I put it. This is the reason why I think you guys should have coding, because I'm like doing it to myself right now. Oh yeah, it's under Scratch. I'm just, sorry, I'm used to working with. I was just gonna show you guys the trim log real quick, just so we can see why we run FastQC again. Because the trim log will actually give you information on like what the program actually did, like as far as like what it actually looked, what sequence were like surviving, what sequence did it chop off. But you can see, once I show it to you, that again, similar to looking at the actual raw FASTQ and trying to decide whether or not that FRET scoring, whether or not you have good or bad quality. Here, trying to understand whether or not you're trimming like what you want to trim can be, you can see here, it's a little, it's just very difficult to kind of read. I mean, at least I can't read it. So that's why after we run Trim-O-Matic, typically what you do afterward is you'll run FastQC again, and then you'll just compare them side by side and see if it actually trimmed what you expected to trim. So that's what we're gonna run. So one other thing I wanna bring up is, again, as I mentioned, like if you look into the data file, you'll see that there's an N1 and an N2. Here we're working with pair N data. Sometimes you're gonna have single N, so Trim-O-Matic pays attention to that. So you'll wanna make sure that you're specifying in your code whether or not it's single N or pair N. So I think here I have real quick for you guys. Like I'm showing you here for pair N, for example, you want to include both. You want to have it point to both the FASTQ for the N1 and N2. And then you want to tell it that when it comes to Illumina clip, to specifically look for the pair N adapters, right? But when you do single N, it's going to be a little bit different. I think I actually have it in here. Or maybe I don't. We can look at the website real quick. When you look at single N, then the parameters and what you actually have in your script to specify that, it's going to be a little different. So here is the actual website for Trim-O-Matic, and you can see it tells you if you're single N, you're going to specify it as SE for single N, and it's telling you that you want to give it what FRET is, but then you have one input. Whereas pair N, which is what we're working with today, you'll see that it has input 1 and input 2. So these are going to be your N1 and your N2. And then you're going to have it output that out, and then it's going to actually combine together in the end. So it's going to, when you look in your output folder for Trim-O-Matic, you'll see that you're going to have multiple files because it's going to try to trim both of them and take into consideration that they're pair N. Does that make sense? So if you open up your script, I'll open up this Word document real quick, and we can just kind of work through it similar to FASTQ. Yeah, I think let's set a time, because I think we're going to do this, and then you're going to run a FASTQ, and then I think we're going to go into mapping. So when you're done, let's just find a time to start again. So let's see, this is, how long, maybe like, start again 20 minutes? Is that the right time? 10:50? So let's come back. So if you happen to finish early, let's just make sure that we're back here by 11:15. Does that sound good? That would be like yeah.
Have you enough time for mapping?
Yeah, I think the mapping, I think that we also have them doing the same file for mapping for homework, right?
Yes,
So
if you don't finish, then you can just keep going and do this for homework.
Okay,
Because
yeah.
I think the trimming is the same files too.
Yeah, the trimming is the same file. We're going to keep moving with these files, and I think we're moving on with them for next week. So if you don't finish, come to the office hours, or or just do it for, you can finish it for homework. So let's just make sure we regroup at like 11:15, and then Jane's going to go over mapping, and then I think the thing that we really want to do is just trying to make sure that everybody can get it onto X to go, so that you can do your homework, and then you'll be ready for it. That's kind of. So there's a worksheet that you can work through similar to the FASTQ that goes over Trim-O-Matic, and there's the links there to the manual and everything like that. We'll kind of walk around just after you get the FASTQ.
Reiterate what they're supposed to be doing?
So right now, you should be, so you have the file, you looked at the original file and you saw whether or not you wanted to trim adapters. We're gonna go ahead and see we want to trim adapters for it. So you're gonna use Trim-O-Matic. There's a S-Batch code in there that is specific that has Trim-O-Matic. You wanna trim the adapters and then you're gonna wanna run FastPC again and just make sure that you're getting back the results that you are hoping to get after you remove the adapter. So that, and then that'll be our stopping point for until 11:15, in which case we'll take those trim files, because it'll give you the trim, you'll get another FASTQ as the output. So your output will be trim FASTQ for both of them, so you should have two trim FASTQ files for N1 and N2, and as well as the FastPC HTML output. And we'll take those same trim and move forward after for the next section, part 2, where we'll actually map them. Is there any questions? I know it's a lot, like all of a sudden there's a lot of data and there's a lot of different softwares. We'll be around, but is there any specific questions on what we're doing? Yeah. I mean, you can keep building if you want to.
The trade-off on that is that if you have a really long S-Batch script that has a bunch of different programs, then it's harder to troubleshoot. Okay. But then if you have a bunch of different ones, then you gotta keep track of them. It's just kind of a trade-off, whatever makes sense.
We're trimming the same file over.
Yes, but you're gonna pull in both those, because remember I mentioned that they're parents. So you're trimming them, but before with FastPC you just did one of the files. Now you want to do both of the files, because we're going to Trim-O-Matic is going to take into consideration the parent, and then therefore you want to FastQ see on both files as well, just to make sure that they're good enough quality you want to both files. All right, so yes, we'll get started. We'll walk around.
Like, it doesn't really matter where you put it. Yeah, I mean, like, if I was saying myself, all my error, all my dot error dot outs on my thing, and all my outputs. Yeah, or you, whatever you're. So it's not, you can be
I see, so you're, yeah, so I guess if you're gonna LS,
that's the two. You have to obviously start to put it.
that's fine.
So if I were to just go down here, slash. That's. So it's best to just.
Where's your out?
Bless you.
Yes, there it is.
Look at that, two sides. There you go. Yeah, fix your directory.
So at least some people are having a problem with FastQC module 0.1.8. So you might want to go back to 0.11.5. I'm not sure what's up with that, but this is up.
I don't know, either, I don't know. Others are really,
So much what was the original script?
Oh, it is.
And people are getting FastQC. So in your trimmatic script, it loads FastQC 0.11.8. Change that to 0.11.5.
Thanks, Lynn.
With this, it's mostly just changing the directory, right? Yeah, I got that to compute.
Yeah, so you copy your path.
What's the version of Trimmatic?
Trimmatic, I don't know, I've had it with one of those lines. I don't know, I've had yes.
Yeah. Hey, everyone, since we're kind of like running a little bit behind the schedule, I kind of want to just like go over the concept of the next section, which is the read mapping, really briefly. And then don't worry if you're kind of a little bit behind. We will spend the entire Friday to really catch up, to go over all the things that we covered today, step by step tomorrow as well. All right, so today we're really just like, I'm really just hoping to cover some of the concepts that involves in the very next step, the read mapping. So to kind of like really, this is like a simplified version of the whole pipeline that we are covering today. So what Jessica has covered really is how do we convert the raw reads into the trimmed reads, and then doing the FastQC to evaluate the sequencing quality before and after the read trimming. So the very next step we're going to need to do is kind of the read mapping. This is really the essential step for us to generate reads that are analysis ready. So, and then if we have time, we probably don't today, and then so maybe tomorrow I'll also go over how do we visualize this kind of like the data. So far we have only been handling like the text files. So eventually I'll show you how to really use the software to visualize the reads being mapped onto genomes. All right, so when we talk about read mapping, what do we really mean? So what we get from the sequencer is really a set of reads, and then the target for us to do is the objective is really to map those reads and then really locate where they're from in the reference genome. So in this case, today we're going to be handling this, as you might have already noticed, the RNA-seq reads, and then the reference genome is the human. And then so we are really talking about a set of reads that are over millions of them, and then we're trying to map them onto a reference genome that's close to 6 billion base pairs. So how do we do that efficiently? So thankfully, there are already like software engineers that like devise different software algorithms to carry out this mapping. So, and then when we talk about mapping, it's very similar to what we are more familiar with, is kind of like the alignment of sequences. So we find in the reference genome a section of the sequence, and then we pull out one read and then see how accurate does this one read map back onto the reference. So this is really, and then as you can imagine, the read might have some mismatches, and then the read might have some insertions, and then those are all taken into account when we do this kind of read mapping. And so overall, what we have since really the first introduction of like the next-gen sequencing, we really, there are like over hundreds of different read mappers, and then really which one do we really choose from out of all these read mappers? So to choose between different read mapping softwares, we need to take into account what kind of sequencing we're doing. So in the case of RNA-seq, what we know is that for mammalian cells, for eukaryote cells, the RNA goes through post-transcriptional modification that involves splicing of the introns. So a read mapper would be able to, in order to map an RNA-seq back into a reference genome, which is the DNA, we will need to make sure that the read mapper is aware of some of the RNA splicing. On the other hand, methylation sequencing, or like any kind of sequencing that involves sequencing some of the reads that are already post-transcriptionally edited, meaning sometimes in the past I have done like double-stranded RNA sequencing. So the double-stranded RNA would go through a base pair of A to I editing, and then in using a typical read mapping software, they would count that as a read mismatch. So and then this is the instance where you might want to relax some of the parameters when you do read mapping, so that they take into account some of the RNA modifications. And then there's also when you're trying to figure out especially the genomic variance between say one individual from another individual, this is where some other kind of like sequencing read mapping software comes in, where they can really be more tolerant of like the variance between DNA sequences or SNPs in this case. So for the purpose of this tutorial, we're going to be focusing on the RNA-seq, and then that's why we chose this really popular read mapping software, HiSat2. So HiSat2 is short for hierarchical indexing for splice alignment of transcripts. So it's a mouthful, and then it's really, it's about mapping. So basically what it means is really it first indexes the genome. So when we're talking about index, it's more of a computational term. You can imagine as we are looking for one specific sequence out of the 6 billion genome, as a base pair genome, and then first it creates a library where each segment of the genome is stored with the indexing. And then so basically, so you first create an index of our reference genome, and then followed by using this splice-aware algorithm that really map RNA reads back to a DNA genome. So to do that, in the first case, when we get a RNA sequencing read that completely perfectly match back onto an exon, it has no problem. So in this case, it's an easy case where we are simply aligning a read back to the exon. And then in the case where there is the read spans a exon junction, what I'll do is first it finds where this read anchors the best, where it matches the best into the genome, and then it kind of like allows or tolerate this kind of mismatch until, and then further extends over the span of an intron, and then eventually find matched reads along the other exon. So in this case, it tolerates the intron and then allowed a intron-aware read mapping. And so this is the software we're going to use. And then without further ado, I would like to first go over the input file that we need for HiSat2. So like I mentioned, HiSat2 uses genome index. So it's an indexed genome to allow faster lookup. And then as well as the trimmed reads that some of you might have already generated. So to do this, let's go to, you would want to kind of download first these worksheets that I have uploaded onto our website. So go to short read and then go to day 4 and then click on this mapping and IGV worksheets. All right, so once we have this worksheet, and then, so basically what we're going to be doing will be very tightly following this, the pipeline that's outlined in this worksheet. And then, so next, let's go over to the terminal. So I realized in the previous hour, everybody has been having a lot of issue really just trying to find the files, or then like trying to locate where their output file is. So I would highly recommend that everybody just like stick to the same path for this section of the tutorial. So, and then where we will kind of just go through this step by step. All right, so the first thing we want to do is to, still up there. All right. Here we go. So yeah, so the first thing we want to do is let's just follow command by command. So in this case, let's go using the tilde, go back to your user directory. So your directory right now should be following users and then followed by your username. And then if you have not already done that, make a directory called day 4. So short read 2021 and day 4. So since I already have this directory, it'll say it exists. All right. Everybody's with me? Great. So now in the day 4, let's first create a directory called HiSat2. And then also make a directory called EL files, like we have done 1,000 times. Since I already have that, it would say file exists. And then also, at the same time, we want a directory to store our scripts. All right, so now if you check your directory, you should at least have EL files, HiSat2, and scripts. Okay, so now let's go into the script. And then here, let's create a file. Let's copy a file from this repository that we have for you. Scratch public, share public, short read, and do script, and D4, and D4. So now we need to copy this Scratch shares from Scratch shares public short read scripts day4 and day4 mapping to our script directory. And then put up your red notes whenever you run into problem during this process. All right. So now you should have this day4 mapping as batch inside of your script directory. Can I get a show of hands to see how many people have gotten this step? All right, great. So now we have this sbatch file, and then really all we need to do is editing this sbatch to run all of the rest of the programs. So like we've done before, let's vim into the sbatch scripts. All right. So you might notice this script is way longer as there's like several commands that we will go through. So start from the beginning. Let's edit this script to make sure everything's there. So job name, day4 mapping, mail user. Email. All right. And leave everything else the same. And then this is really a lot of, I see a major roadblock for a lot of people. It's really where we have these EL files. So like since we have just created this directory, we want to just make sure that directory specified is correct. And then we can copy and paste this. Okay, let's focus on the header for now. And then you can put up your green sticky notes once you finish your header. This is the mapping. Good point. Make sure the partition is not short. It's compute. Right. And then you can always put up your red sticky note in case if you can't find the files or run into problem. Oh, okay. Gotcha. I see. All right. All right, so far, so, yep. Yeah, the copying the file from, oh, I see. So, let me create a new one. It'll be from, let me type it here. So it'll be from this. Oh. All right, yep. Any questions so far? All right, so seems like a majority of people have gone to this step so far. All right, so the next thing we need to do, so basically the rest of the section, the next section remains the same. The next thing we want to do is to specify where the trimmed reads are and also where the reference genome index is. So to find those,
Shane, can you move it so that it's a little bit bright?
uh,
Left is cut off.
oh, how do I, oh, let me, okay. Yep. All right, so first we need to find out where those genome file is. So if we do ls, and the genome file location is actually quite long, so I would highly encourage you to use the tab, and then this will be the genome file that we use. All right, so what I'm going to do is just simply copy this to our script, and then specify a variable name for it. So in this case, it'll be called genome. And then we can copy and paste that path we just copied. All right? And then since we also don't know, we'll come back to this, so don't worry if you haven't finished typing it down. The next thing we need to copy is the trimmed reads. I noticed that some of you might not have gotten your own version of the trimmed reads. But I have already prepared the backup version of it. So to get those, what we can do is go to Scratch, Shared Public, Short Read, Cooking Show. And this is really where all of your answer is. And then day 4, and then followed by Trim-O-Matic. And then so all right. So here we go. The 2 the trimmed reads that we will need is going to be stored within this directory. So let's copy this one over as well. Okay. And then we name them as read 1, read 2. All right, so make sure we have everything from here, the genome, the read 1, and read 2. And don't worry about the other parameters. We won't need them. Oh, actually. Is everybody following? All right. Can I get a show of hands of how many people have finished typing down? Or green stickies? Okay, so far what we have, sorry, so the other thing we do want to keep is really the output folder. So this is where we're going to be storing all of our mapped reads. So that's not correct, sorry. So this will be the same directory that we created. So for the HiSat2 will be the output directory that we are going to store all the mapped reads. So this one will be users followed by your username, short read 2021, day 4, and HiSat2. And this is the directory we have created earlier. Okay. So really, to map the reads, these are the 4 key things that we need. Reference genome, the pair end reads, read 1 and read 2, and also the output directory called hiset2. Okay. Correct. Yeah, yeah. Make sure, yeah, it's. Oh, that's correct. I put it as a lowercase hiset2. Great.
Our script already had.
Right, so that one was more tailored for the Fiji server. I wouldn't use that, but instead, since we have just created this user's username, and then in day 4 we have a direct hiset2, let's all use that one. So we are all in the same, so on the same page. Okay, so now we have these 4 variables. Let's go to the next section. Again, it's module load. So over here, we will primarily use this hiset2. The SAM tool will be the next step. I will come back to this in a little bit, but then just leave this section as is. I will then later go over what each tool does. So now to run the pipeline, let's go over to this hiset2 command. We're going to need to edit this a lot. So let me first just go over what these are. So going back to this presentation, to run hiset2, what we need is really just this command with typically 1, 2, 3, 4, 5, 6 lines. So we start with calling this program hiset2. And then what's really nice for hiset2 is it has some default parameters to allow you to whether, if you want to map the reads really fast, you can specify very fast. If you want to map the reads more accurate, you can specify very sensitive. Whereas, and then it followed, same as other programs, it follows with a flag. So in this case, the X means the index. So followed by the genome index that we created as a variable, as well as the trim the reads. So in our scripts, we specify those as read 1 and read 2, and then followed by the output. So output for hiset2 uses a forward arrow. So in Unix command, this simply saves everything the program output into a file. And then followed by that, the next line would be 2, followed by a right pointing arrow. And then this is also a universal Unix command to save all of the arrow file or additional output file that this program generates. So basically through this total of 6 flags, we tell hiset2 to run in a specific mode, tell them what to map to and what they use to map, and then also the outputs. All right, so now we go back to the script to kind of edit this hiset2 command, which is right here. All right, so since we have already saved everything into variables, let's make it faster by yeah, exactly. We just call the variables. It might be faster to just really retype the command and call the variables. So again, kind of following this existing command, what we have is very fast. Oh, whoops. All right, here we go. All right, so let's just retype the command since we already have the variables. So again, it starts with hiset2, very fast. And then the first flag we're going to use is -x. So in this case, we're going to specify our genome. This is the genome index that we specified as a variable. And then as a side note, the bracket that we used to point out a variable is sometimes optional, especially in the case when you are have this very well separated commands. But then sometimes the program might get confused of what's the variable. And then so as a general best practice, you would use the bracket, but then sometimes you can delete it. It still works. And then we'll have read1 followed by the -1 flag and -2 flag, read2, and then followed by the output. So for output, we could really use what's already specified. The copy paste doesn't work. 2 followed by, so here let's just call it 21 Eric.sam. And then last thing is some of the additional output coming from this HiSet2. And then we can again call it chromosome 21 Eric dot HiSet2 map stats dot. All right, so that's really all you need to run HiSet2. No, not yet. But you can definitely aspect right now to get the SAM and the map stat. Yeah. But then given the 10 minutes we have, let's hold on to that and then come to our office hour when you aspect this or submit this job script and then run into any error. But then for now, as long as you have this along with your variables specified correctly, you should be able to run this. All right. Okay, so now we should have everything in the view. So yeah, make sure everything in this entire section is in your job script. All right, for the time being, let's just pretend that we all submit the job script and you ran, and then we have our output. What we are going to end up getting, like we specified, will be a SAM file and then a map.stat file. It's a text file. So if we actually dive into the SAM file, what we're going to see, let me go back to here, is basically for every single read, oh, the animation's not showing up. All right. So it's basically, the SAM file is also a text file that has like millions of lines of text that looks kind of like this. And what we're going to get in this SAM file, that the information that's captured in this SAM file starts with a Q name. So those are all the variable names the column names for each variable within the SAM file. And then I'm just going to go over what each one of this means, because for some of you, if you're doing certain kind of analysis, this might be the terminal file that you need. So to look at this, it starts with a read name. So this is really the name of that one specific read. And then followed by a flag. So the flag here tells you really a lot of info. So in the case over here, it says 16, but then it could also be other numbers. It really specifies whether a read is mapped or not, or whether how this read is mapped onto the reference. And then followed by that, there's also the where it is mapped specifically. So there will be chromosome name, the position of where on that chromosome is this read mapped to, and also the mapping quality. So this is like a generic score that summarizes whether this read is mapped well or not. And then followed by this cigar. So cigar is rather a sequence of letters that kind of represents what you have to do to these reads in order for these reads to be mapped. Like I mentioned, sometimes you'll find mismatches. So you might need to change one base pair in order for this read to map. So, and then those kind of information will be stored into this cigar variable. And then you can definitely look into more of this manual of the SAM file, and then to get a sense of what kind of modification that this variable can specify. And then the next three variables, it really is kind of, so in this example, we're actually showing us a single AND reads instead of a pair AND reads. But then really these three variables here really specifies if in the case of a pair AND reads, when the reads is paired, where is the other pair of this read? And then very similar to FASTQ, the next line specifies the sequence of this read and then the quality of each individual read being mapped onto the reference genome. All right, and then finally, remember we also created a second output from the HiSat2 command and as a text file, and then we named it as HiSat2 mapstat. And so within this text file, this is where you will get a lot of the useful overall information of like how is this mapping performance. So for typical RNA-seq reads, if you found if your sequence doesn't have any contaminations, and then if you are using if your reference genome is robust, then you would expect an overall above 90% of the read alignments, meaning that 90% of the reads that are used for inputs are ended up mapped onto the reference genome. So now, basically, this is the two output that we will get from read mapping, and then with the SAM file, the very next thing will be kind of converting it into a more condensed and also machine-readable file called BAM file, so the B for binary, and then that's achieved through the next three commands that's also included in the script. And so I would encourage you to, since we're getting close to the end, I would encourage you to kind of follow the worksheets, and then you might need to change the input or output directories based on where your files are. And then we will all be here, all the helpers will all be here to help you navigate those paths. And then, so the end goal here is that we basically convert the SAM file into a BAM file through this command. So using the second software that we module loaded called SAM tools. So the first thing we'll do is converting the SAM to BAM. So this is achieved through this SAM tools view. It's kind of weird that they call this conversion command as view, but then followed by the number of CPUs that you want to use, followed by the file format as the input, and then the output BAM file. So, and then followed by, once we have generated the BAM file, the original default order of this BAM file of every single read is really rather random. We want to sort them based on the chromosomes as well as the location of the chromosomes, and that's why we need to sort the BAM file. So, so this is achieved by SAM tool sort, and then again, you just put in the input file. In this case, it's the converted BAM, and then output it as a sorted BAM. and then eventually you want to tell whatever the program that you're about to use next that what kind of chromosome format this band file is in. This is achieved through this SAM2 index, and then you'll end up getting another index file where we specify the suffix as the BAI. And over here, if you take a look eventually at this BAI file, what you'll get is kind of the chromosomes and then really how long each chromosome is, and then the total number of chromosomes that was used for mapping. So that's really all we need to cover today. And then don't worry if you are feeling kind of like falling behind or getting lost with all of this path in, path out. You can either stay for the afternoon where we can like troubleshoot and then navigate through to generate these files, or tomorrow we'll also have a more systematic walkthrough of this entire process, followed by some of the questions or quiz that you have to answer. Questions so far? Right, so basically, like Robin said, mapping is really the very last step. We need to, the very minimum last step that we have to do before we have this analysis ready mapped reads that you can be used for either finding differentially expressed genes or finding variants within genomes. Okay. All right, I think it's about time for this morning session. So like I said, stick around. I know I didn't leave any time to kind of practice. So stick around or come back tomorrow, and then we'll go through how to write the script accurately.
If you're able to try to get X2Go working, if you haven't already, that would be great before tomorrow too. And also, there's a new worksheet. So under day one, there were X2Go instructions. There's a new worksheet under, I guess, PDF under there for Windows users, just to make sure that the authentication is okay.
From what we did just now, would we get?
Just the SAM file, and then
Okay, my
right.
SAM was empty, so I didn't pick wrong one.
Yeah, I guess.
Yeah.
Yeah, I think, yeah, regardless, since it's like since you use the Unix for design, you get I was trying out this new method, but then I felt like still
I don't know
advice
which one
at
is
different
better,
pace.
like to go slowly.
Yeah.
Have everyone follow, because then I feel like it takes so much time. But then I don't know if like having to just do it better.
Right.
That also
I
seems
know.
to create chaos. It takes time.
Yeah.
So I don't know. Yeah, I was debating it like last night. I was like, well, should I go through step by step,
Yeah.
or should I like just give them the worksheet and be like, have at it? I don't know.
And then the hard thing is like we prepare all those worksheets for Fiji. So
Yeah.
now all the paths are incorrect and everything. I might remake my worksheet for tomorrow so that like everybody just like create